Hi, my name is Tomohiro Morio at TMDU.
Hello, my name is Motoi Yamashita at TMDU.
Today, we'd like to talk about a recent study on a novel immunodeficiency. Inborn errors of immunity, IEI for short, are a group of more than 400 genetic disorders in which part of the body's immune system is missing or functions improperly. IEI manifests as increased susceptibility to infectious diseases, autoimmunity, autoinflammatory diseases, allergy, and/or malignancy. Our group at TMDU has been elucidating molecular pathogenesis of these disorders and has recently identified a novel disease caused by a mutation in IKZF3 gene, AIOLOS gene.
We studied a family of patients with B-cell deficiency. A heterozygous missense mutation in the AIOLOS gene was identified by whole exome sequencing. AIOLOS belongs to a transcription factor family called IKZF. IKAROS, the pioneer protein of the family, is already known to be the cause of IEI, which we have reported a few years ago. But AIOLOS deficiency was not known then. IKZF molecules form dimers to function as transcription factor complex and regulate the development of lymphocytes. Structural analysis predicted that the mutated AIOLOS protein loses DNA binding ability. In vitro analysis supported this hypothesis, as shown by loss of dot pattern representing normal DNA bindings. To further study molecular mechanism of AIOLOS deficiency, we generated mouse having the patient's AIOLOS mutation. The patient model mouse showed reduction in the B-cell number as observed in the patients. There was a differential blockade at pre-pro B-cell stage to pre-B-cell stage, where the AIOLOS starts to express during the B-cell differentiation. Additional studies suggested that the mutant AIOLOS interferes with the function of IKAROS by forming heterodimers. The mutant AIOLOS prevents IKAROS binding to the physiological transcriptional regulatory regions. Also, IKAROS were taken to non-physiological binding sites by the mutant AIOLOS. We showed that the dysfunction of IKAROS, rather than that of AIOLOS, is responsible for immune defects observed in the patient model mouse. Finally, to seek therapeutic potential for the AIOLOS deficient patients, we performed gene therapy in the patient model mouse. Additional mutation that truncates protein interacting domain of the mutant AIOLOS was introduced to the patient model mouse. If function of IKAROS is compromised by mutant AIOLOS via formation of heterodimer, deletion of dimerization domain would restore IKAROS function and prevent immune defects in the patient model mouse. As expected, the gene therapy mouse recovered the B-cell number.
Our detailed study led to identification of novel molecular pathogenic mechanism, which we named heteromeric interference. Many proteins in a body function together as protein complex. Thus, it is highly likely that a variant of one molecule could interfere with its normal partner protein. We would like to point out that heteromeric interference could be involved in other disease processes, such as cancer development and endocrinological disorders. We are hoping to continue to strive for a better understanding of the pathogenesis and for the development of new and better therapies for inborn errors of immunity.